Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending. I'm your moderator, Victoria Rees, editor of Drug Target Review. Today's speakers will be Dr. Celine Legros, Drug Discovery Partnerships Director at Eurofins Discovery Services, and Dr. Philippe Dupois, High Throughput Screening Study Director at Eurofins Rep. Following their presentation, we'll be moving on to a live question and answer session where you can pose your questions to today's speakers. Please remember that you can submit your questions at any point during the webinar using the questions panel, which is situated in the menu on the right. So without any further hesitation, may I pass over to today's keynote speakers.
Hello everyone, Philippe and I are very pleased to present you today this high throughput screening cascade designed to identify novel and tractable hit series of PIM3 inhibitors. Hit finding and lead generation are critical in the drug discovery process. It has to be done the fastest and the most efficiently possible. The perfect lead compound has to be novel, patentable, and show the desired potency and appropriate selectivity, and have an ADME tox profile suggesting good pharmacokinetics properties. Having this perfect compound requires to access a wide range of technologies and expertise, as the chemistry, considering synthetic and organic chemistry, but also computational and virtual screening capabilities, and also for the HTS, access to valuable compound libraries. It also requires strong biology and pharmacology skills, and all of this supported by strong project management capabilities to ensure a smooth, fast, and efficient project. At Eurofins Discovery, we have more than 40 years of experience in drug discovery and preclinical development. From the origin of Eurofins Discovery Services, we deliver more than 30 billion of data points to our customers, related to more of 4,500 off-the-shelf assays. A part of them are enabled for HTS to screen our library of more than 600,000 compounds. At Eurofins Discovery, as you may know, we are able to offer you a full support for your drug discovery project, from chemistry to in vitro pharmacology and safety, or ADME and in vivo studies. And all of those activities can be supported by our Discovery One team and integrated drug discovery services to help you in the design and the implementation and execution of your project. When screeners are thinking about HTS, we think about robots and integrated systems, and some of the HTS platforms are really huge. But depending on the assay used as the primary assay of the HTS, fully automated system may not be required or the most appropriate ones. There are different levels of automation. We can use standard system connected with stakers as peristaltic dispenser or multimodal readers. This semi-automated design represents the advantage of a very high throughput. Basically, you can dispense a plate per minute and having stacks of 50 plates. Unfortunately, some assays, due to their design or their complexity, or because you have to run it 24 hours a day, will require a fully integrated system with a robot arm. At Eurofins, our automation team has access to all of those levels of automation, and they will ensure that when they design the assay and the assay automation, they select the best process for your project. Kinase drug discovery efforts can be particularly challenging due to the conservation of active sites and high structural homology between kinases, often resulting in hit compounds with embedded safety risk or potentially suboptimal success rates in HTS campaigns. So we have access to a comprehensive kinase toolbox at Eurofins Discovery that will support your project from all along the preclinical phases. We have more than 20 years of experience in kinases, with more than 420 kinases in our catalog and ready for testing. Our kinase offer is mainly supported by two complementary platforms, a binding one, the KinomeScan, and an activity-based one, the Kinase Profiler. As the complexity of the kinase drug discovery can't be addressed only with those two platforms, we are also offer multiple assay formats and technologies for an efficient HTS or E2 programs. This includes mass spectrometry, standard biophysics technologies, also cellular target engagement assays as in cell hunter or NanoBRETT technology. You may be wondering why we selected the PIM3 kinase. The PIM kinase have been first identified as pro-viral integration site for the mononamed murine leukemia virus. The PIM family is composed of 3 serine-threonine kinases that are PIM1, PIM2, and PIM3. This kinase subfamily has the particularity to not have a regulatory domain that made them constitutively active. And they are a critical step in the ABL, JAK2, and FLT3 pathway. From a therapeutic perspective, the overexpression of the PIM3 has been reported in some tumors and cancers that make those kinases very attractive target for cancer therapies. I will now present you the screening cascade we design and execute to identify new PIM3 inhibitors. The primary assay of the HTS is the ADP glow assay. In this assay, we use the protein produced by our production protein team, EDPF, and we screen a portion of our library, about 70,000 compounds. We follow the classical pathway for HTS. It means pilot screen, one dose screening at 10 micromolar, and dose response curve confirmation. Following this primary assay, selected hit will be characterized in dose response curve in 2 PIM3 orthogonal assays. One level 3 mass spectrometry assay, and the second, the NanoBRETT cellular target engagement assay. Following the orthogonal assay, we will investigate the selectivity of the confirmed hits for PIM3 versus PIM1 and PIM2, using both platforms, the kinase profiler and the kinome scan. If needed, we could also extend the selectivity panel to the whole kinome using the same platforms. The final step that won't be presented today as the study are still ongoing is the investigation of the mechanism of action of the compounds using biophysics methods, and also the characterization of the compounds in relevant oncology cellular models on our OncoPanel platform. Now Philippe will present you all of the experimental results we have obtained during this project.
Yes, so thank you, Céline, for introducing all this. I will now introduce how we executed this work, and I will share with you the key figures in terms of results and data analysis. So as I indicated, for the primary screen, we use the ADP glow technology from Promega. The 71K compounds subset of Eurofins library was prepared into assay-ready plate using an eco nano dispenser. And as Céline said, the recombinant active PIM3 kinase was produced in-house, which was a great advantage. As you know, Eurofins has a great expertise to develop assays. We did it thousands of times over the 30 last years. I will not go into much details, but we optimized the enzyme and substrate concentration, the co-substrate concentration, and determine the key M value for each of them. We also work on buffers, their pH, ionic strengths, and components. We optimize incubation times and temperature as well, and we determine the upper limit in DMSO as a final percent. Eurofins' strength is also its ability to miniaturize and industrialize the assay to make them high throughput compatible. As Céline previously described, we indeed adapt the level of automation to each project to be on top of efficiency and robustness. Here, we use via fill and also multi-drop dispensing. We add, in terms of protocol, we add the enzyme and the peptide substrate onto the ready assay plate containing the compounds and controls, and start the reaction immediately by adding the ATP. After 40 minutes incubation at room temperature, we add the ADP glow detection reagent, and after 40 more minutes, we add the kinase detection reagent. The plate signal is measured after 30 minutes using a Ferrastar. So when it's for clients, we provide a full assay development report with all the details and the SOPs. Once the assay is developed, We first start, of course, by a pilot screen to challenge the robustness of the assay and also to predict the hit rate of active compounds. For this purpose, the pilot screen was performed using 20 plates randomly selected from Eurofins Discovery Diversity Library. This represents a bit more than 7,000 compounds. They were tested in silicate at one concentration of 10 micromolar in 2 independent experiments. As you can see, the signal windows was pretty comfortable with the SNR around 40 and the Z prime factor over 0.8. And this was true for both individual experiments. So SNR and Z prime were stable and above the standard. When we look at the correlation between the 2 independent experiments, we can see that there is a perfect correlation between the percent inhibition obtained in the first and the second experiments. The assay is repeatable and robust. We also display the Venn diagram, which is another way to represent the things. On this last graph, we report the distribution of the activity of the test compound. Each bar represents the number of compounds which display the given percent of inhibition from the active on the left to the more active onto the right of the slide. We observe a Gaussian-like distribution. We can then calculate the mean activity value. We can also calculate the standard deviation, and when we apply the mean plus 3 standard deviation, then we define the selection threshold to apply. It means that any compound above this threshold can be considered as an active compound. With this low selection threshold, the hit rate is estimated to be close to 1.32%. This analysis and reported is indeed the kind of information we share with our clients when we perform such a project for them. So the pilot screen gave us a clear go for the one-dose screen on our 71,000 compound library. They were tested in silicate at one concentration also of 10 micromolar final, and this time at a single experiment. As expected, the signal was still comfortable with the SNR around 35 and the Z prime factor still over 0.8, and even a little bit better than the pilot. So the assay performance perfectly matched with the pilot. When we look at the distribution of the activity of the test compound, we observe a Gaussian-like distribution, like for the pilot. And we calculated the selection threshold of 31. So any compound which displays more than 31% inhibition can be considered as active. And based on this, we selected 1,000 active compounds, which correspond to a hit rate of 1.54%. The hit rate is a little bit higher with the 70K compound set compared to the pilot screen set of compound. This is due to the fact that the 71K compound set is kinase focused. As a conclusion, the one-dose screen was as good as the pilot screen. The results were perfectly in line with the pilot screen data, and 1,000 compounds were selected for dose response curve. So let's have a look to the dose response curve results and analysis. In order to determine the IC50 and the PIC50 value, we ran dose response curve. It means that the 1,000 compounds have been tested in duplicate at 8 concentration in 2 independent experiments. The signal windows was maintained with a SNR around 27 and a Z prime factor over 0.8. And here again, it was true for both experiments. The dose response curves were robust with high quality like in previous steps. When we look at the correlation between the 2 independent experiments, we see a perfect correlation, so we can trust the hits we selected. If we report compound PIC50 frequency of distribution, we can say that we determine an IC50 value for 946 compounds over the 1,000 selected and tested, and we get PIC50 from 3.4 to above 7.5. So it means that the most potent are in the 10 nanomolar range, which is not so bad indeed. Once again, it was a pretty successful step with 94.6% confirmation of the effect. With the help of our chemists, based on the potency and the scaffolds, 140 compounds were selected for orthogonal assay. Moving to orthogonal assay, as introduced earlier by Céline, they were indeed based on the rapid-fire mass spectrometry model as an activity assay, and NanoBRETT as a cellular target engagement assay. Here again, we fully developed and optimized these two models. I will not go into much details here. The two protocols are displayed on the slide, and the key points are that we still use our libraries, the nano dispensing, the semi-automated workstation to reach a pretty good efficiency. The readers were the Q-trap for mass spec and Ferrastar for fluorescence. In both models, the 140 compounds were tested in duplicate at 8 concentration in 2 independent experiments. Concerning the rapid-fire mass spec activity assay, the signal window was comfortable and the Z prime factor still acceptable, still above 0.5 indeed, and even if a little bit lower than the previous steps. If we look to the graph on the left of the slide, we can see that the correlation between the two independent experiments in mass spec is very good. If we look to the graph a little bit on the right side, we can see that the correlation between the ADP glow and the mass spec results were pretty good also. 95.7% of the hits were confirmed in mass spec compared to the ADP glow. However, we observed a shift of 0.77 if we consider the PIC50 values, which mean that compounds are alpha log to 1 log less potent in mass spec than in ADP glow. Concerning the NanoBRETT intracellular kinase assay, the signal windows was comfortable also and the Z prime factor pretty good at 0.83. If we look into the graph on the left, here again, we can see that the correlation between the two independent experiments in NanoBRETT is very good. And if we look to the graph on the right side, we can see that the correlation between the ADP glow and the NanoBRETT is good also. We are here in a whole-cell model, and not surprisingly, we observe a shift of 1.24 if we consider the PIC50 value, which means that compounds are 1 log to 1 log and a half less potent in NanoBRETT than in ADP glow. This explains why 47.9% of the hits were confirmed in NanoBRETT compared to ADP glow. As a conclusion, for the two orthogonal assay screen steps, we can say that we had a very good correlation between the two experiments and the two technologies. And one more time, with the help of the chemist, 16 hits over the 140 hits distributed in 6 clusters have been selected for the following steps. Further triaging between the 16 hits took place employing Eurofins Discovery proprietary kinase profiler activity platform to investigate their selectivity against PIM1, PIM2, and PIM3 kinases. This panel is part of our standard offer. You can find all the information on the Eurofins Discovery website, and we run this panel in routine on our site. The assay principle is described on this scheme. It is a radiometric assay, and it is an activity-based platform. The correlation between the ATP glow and the kinase profiler results is good. We observe a shift of 1.01 if we consider the PIC50 values, which mean that compounds are 1 log less potent in kinase profiler assays than in ADP glow assay. All the 16 hits were confirmed in this fourth activity assay with some selectivity for PIM3 kinase, and 2 scaffolds show selectivity for PIM3 versus PIM1 and PIM2, which is very promising results. So if we summarize, we developed several models based on ADP glow, NanoBRETT, and mass spec. We went through the pilot screen, the one-dose screen, the dose response curve, the orthogonal screen, and the selectivity assays. We managed to identify compounds with structures known as PIM3 or kinase inhibitors, but also, very interestingly, with novel scaffold. The non-inhibitors were not selected in clusters. Indeed, from our 71K compounds, we selected 16 hits distributed in 6 clusters with novated scaffold, which are chemically tractable. The hits are chemically tractable if we consider the scaffold and their chemical properties based on their molecular weight, calculated partition coefficient, the presence of hydrogen bond donor, the presence of hydrogen bond acceptor, and also the presence of heavy atom. With the ADME studies for series prioritization for It2Lead program, we are one step further to what we consider to be part of the standard HTS campaign. We indeed took advantage of the full access we had on site to our ADME platform. And we were also in a very comfortable position to have enhanced several clusters and compound series. So it was very exciting for us to try to prioritize them a little bit more. We succeeded to identify 2 chemical series as priority 1 for the future It2Lead program. For this purpose, we indeed ran a small ADME package in order to evaluate the compound solubility in PBS, to access the lipophilicity expressed as the log-D or partition coefficient, and also evaluated the mouse and human hepatocyte stability, which give us the estimation of the interesting clearance of the compound half-life. So I will stop on this very promising view. This is what I wanted to share with you about what we did at the bench for this project. And I think that Céline, you have a few more slides to conclude.
Thank you very much, Philippe. So in conclusion, we have been able to elaborate a robust hit finding strategy that combines multiple assays and readouts, as you could see, to identify novel and selective PIM3 kinase inhibitor scaffold in just a few months. The design and the execution of the screening cascade leads to the selection of 2 chemical series to launch an It2Lead program. Screening cascade that combine protein production, HTS assay development capabilities, compound libraries, and a wide range of technology available at Eurofins Discovery. This study also demonstrates the importance of having access to highly skilled project manager and scientific experts to be successful and effective. So during this webinar, we talk a lot about kinases, but at Eurofins Discovery, we have a large screening and profiling portfolio that address most of the target class, as the GPCR, the ion channel, the nuclear receptors, or even the epigenetic targets. We have experts and expertise in multiple readouts and technologies that are compatible for screening of hundreds of thousands of compounds or for the profiling of your key compounds and technologies as the TRFET, the EFC, the mass spectrometry, or even the biophysics methods. If you are interested in any target for an HTS or an It2Lead leader program, do not hesitate to contact us, and we will be happy to design with you a comprehensive screening cascade and to connect you with our experts of specific technologies or therapeutic areas. So what's coming for the future? The cascade is nearly completed. Some of the most promising hits will be run in the Safety 44 panel as an early de-risking strategy. As Philippe highlighted, we are in a very comfortable position with multiple scaffolds possible. And also, based on the data and what Philippe already mentioned, we decided to initiate a small It2Lead program for our most promising and selective hits. And we were expecting to show you the follow-up studies in the coming months. In parallel of this small molecule proof of concept, we were happy to share with you today. We are running a similar fragment-based cascade, including, in addition of these already existing readouts, the binding kinome scan and the SPR single dose screening. I hope to be able to share the whole data set by the end of this year with you in a new webinar. And finally, the next step for the small molecule proof of concept is to compare the gold standard of the ADP GLOW assay for kinases HTS with the HTS mass spectrometry ECO-MS readouts we successfully implemented at CEREP at Eurofins. All of this wouldn't be possible with the support of the whole team at Eurofins Discovery. And thanks a lot for your time, and we will be happy to answer your question.
Thank you, Céline and Philippe, for your excellent presentation. We'll be moving on to our Q&A session in a moment, but just before that, we have some poll questions for you. So the first question for you today is, what are the most relevant target classes for your next hit finding campaigns? Could you just select one out of the below? Fantastic, thank you. So next poll question for you now. What is the average size of compound or fragment libraries used in your high throughput screening projects? If you could just select one of the below again, that would be fantastic. Brilliant, thank you so much for all your answers. And now for our last poll question. Do you have any high throughput screening projects you would like to outsource in the near future? Again, if you could just select one of the below. Fantastic. Thank you so much for your answers there. So we now have the pleasure to introduce our question and answer session, where Celine and Philippe will answer all of your questions. Don't forget that if you do still have any questions, you can still submit these using the questions panel, which is in the menu on the right. So let's take a look at the first question. So Celine and Philippe, in your Cascade, you use the Kinase scan and the profiler platforms. How well do these correlate to find off-target hits?
So thanks for the question. So the Kinase scan is a binding platform, and the Kinase profiler is an activity-based platform. So both platforms give complementary information, and for most of the target of the kinase that's correlate quite well, we have some discrepancy in terms of potency between the two platforms. In terms of profiling of target profiling, it's the same, and we basically catch all the potential of target for kinases. And you can start first with some binding and having some complementary information with the activity platform. I hope I answer your question.
Fantastic, thank you, Celine. Philippe, did you have anything to add to that? Nope, no problem at all. Fantastic. So our next question for you now is, what is the global rejection rate over this high throughput screening campaign? Philippe, I think you're muted.
I was on mute. Yeah, sorry. So I was just saying that I can take this one. Yes. So as you can see, the results were pretty clean, and the rejection rate was quite low. We were lower than 2% in total on the whole cascade. So it was very efficient, very clean indeed. on some assays and some steps, we even had 0 rejection rate. So we are pretty happy with this.
Definitely. So next question for you now. Why is there a shift of potency between various technologies, the ADP Glow versus NanoBRETT versus mass spectrometry versus kinase profiler?
Yeah, so this drives us back to the first question that Céline answered. So we are indeed dealing with different technology, different models, sales models, non-sales models, with also different amount of substrate and sometimes enzyme. We try to keep the protocol as close as possible, but this indeed explained that we could have this shift. What is important is that we always observe the same shift. It means when you correlate the two independent experiments within a single technology, you always confirm your results. And so you can also place or organize the technology one compared to another. You know that you will always have this shift. So this is something it is not surprising, and you can include it in your expected results or shift of potency. I don't know, Céline, you have something to add around this, or?
No. Yeah,
okay.
that's what's important. We keep the ranking of the compound between the different assays. So not a big deal.
Fantastic. And just a reminder that you can still send in your questions, so please do send them in and we'll get them answered. So next question for you now, Céline and Philippe. Do you have any information about the biophysics characteristics of your leads?
So we do not have that much at the moment, but there is a new platform and always new things coming at Eurofins. So we have planned for the further testing on our facilities in Taipei, but also in our facilities in France with SPR technology to come and other possible biophysics characterization. Céline, you want to?
Yeah, the MST trick is coming with the spectral shift as well. So we will challenge this new technology with the PIM3 proof of concept.
Yeah, it will be part of the next steps.
Brilliant, thank you. So what is the interest of multiple readouts for the high throughput screening cascade?
Yeah, I can take this one. From a screening cascade perspective, the interest is that you confirm actually the hits you find in your primary assay and you avoid all the bias using a second biochemical assay, and you are sure the hits you find as inhibitor of your enzyme are real inhibitors. And having a cellular target engagement give second layer of complexity and information in the quality of your hits. It's not mandatory at the end of a HTS screening cascade to have a compound active in the cellular context, but for the kinase, it's give you another criteria to discriminate your compounds, because it's quite easy to find inhibitor of kinases, but have them confirm and also active in the cellular context is quite important. And the advantage with the platform we have, we can easily go through the very close selectivity panels as we did with the PIM1 and PIM2. It's also given priority and higher value to the hits.
Thanks so much, Céline. So how do you secure the supplies during this period of time?
This is a big deal at the moment. So there are, so when we say supplies, I mean, I think that you mean reagents, but also plasticwares and so on. So in terms of reagents, Eurofins Discovery propose a lot of internal reagents. So we prepare and we synthesize and produce a lot of enzyme. It was the case for the PIM3 kinases. We have our own cell culture department. We display some of our ready-to-use kits for some technologies. So we have all this internal production at EDPF that securize and ensure that we are autonomous on this aspect. Regarding the other supply, the plasticwares and so on, we secure everything in terms of duplication of the provider, but also qualification of what we use, from the plates to the tips and so on. So everything is important for HTS campaign. And yes, we have large inventory. We have several suppliers for important reference, and we qualify everything before we launch the project.
Yeah, anything I can add, actually, the fact being a part of the big Eurofins group, Eurofins Discovery give us the opportunity also to collaborate between sites and to transfer some of our stocks to our colleagues who need it. So that ensures business continuity without not too many issues. It's a kind of challenge anyway.
Yeah.
Fantastic. Well, thank you both so much for your excellent presentation and for answering those questions there. Please do join me in thanking Celine and Philippe for their time today. So, as you leave the webinar today, a survey will appear on your screen asking you to rate the webinar. Please do take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. So now, on behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.
Thank you. Goodbye.
Bye-bye.
Bye-bye.
Bye.